Chung Chung, and I'm the Academic Program Manager here at Oregon State University's Hatfield Marine Science Center here in Newport, Oregon. And I, as well as Suzanne, will be your host of today's talk. So a couple of logistics I wanted to share. We have your mics. If everyone could keep their mics and cameras off until the end, we ask that you keep them off for the presentation. Please ask any questions you might have using the chat box found at the bottom or the top of your Zoom screen at any time, and we'll answer any questions at the end of this talk. Also at the end, you can also verbally ask these questions. We will be recording this event, and it will be posted in a few days on the HMSC website on the past seminar page, and I'll post that information in the chat. Just a quick announcement to everyone. Next week's seminar speaker on March 25th will be Celine Derville of the GEM Lab at Oregon State University in the Marine Mammal Institute. And we'll be talking about combining aerial and boat-based surveys to model large whale habitat year-round in Oregon, otherwise known as the OPAL project, O-P-A-L. Again, if you need more info or links to our upcoming events, check out the HMSC homepage and scroll to the bottom for more details at hmsc.oregonstate.edu. And I'll put that also in the chat. So today's speaker has been invited by Susan Brander, Assistant Professor in the Department of Fisheries and Wildlife. And so I'm going to hand it off to Susan for introductions.
Hi everyone, I'm Suzanne Brander. I'm in Fisheries and Wildlife and also the Coastal Oregon Marine Experiment Station. And it's my pleasure to introduce Dr. Samarine Siddiqui today. She is a postdoctoral scholar in our lab and has been with us since last August, although it seems a little bit longer than that because she's really become such a cornerstone of our group, and we're so lucky to have her. She's going to be talking today about some work that she did for her dissertation at Texas A&M, and then may also be chatting a bit about some work that she's been doing since arriving here, which has been on micro and nanoplastic toxicity and also a bit on pesticide toxicity as well. So I'll go ahead and let her take it away, and thank you all for joining us this afternoon.
Hello everyone, I'm Samarine Siddiqui, and as Dr. Brander already introduced me, I'm a postdoctoral scholar at her lab. Thanks for inviting me today. I'm going to talk about contaminants of emerging concern in surface water that includes marine and coastal ecosystem with possible climate change impacts. So first of all, I just wanted to take you from the basics. As you all know, contaminants are called contaminant until they are at the lower level, which doesn't affect anyone. And as soon as they start affecting an organism, they become pollutant. So basically, simply their presence is not making any difference, but as soon as they cross their level, it becomes an issue. So for example, plastics, we found trash all over everywhere, but it doesn't affect until it starts killing or causing health effects to some organisms. For example, in this case, plastic here is not causing anything until we see them actually killing the birds or things. So just to be shortened out, all pollutants are contaminants, but not all contaminants are pollutants. With this, I want to take you through like what are the main pollutants that are in our oceans today. Basically, they are chemicals and trash, if you want to classify them. And just a minute. Okay, so chemicals and trash. Chemicals consists of oil, toxic metals, persistent organic pollutants. Trash means basically we are talking about plastics, and that's the major concern today in terms of pollution. So from where they come from? Most of the time, contaminants come from various sources. It includes forestry, like insecticides and fertilizers used in there. Same with the croplands, industry, livestock, urban development, community parks, they used in their grasses, anti-fouling paints in the ships. Aquaculture is, it's one of the biggest source because it used so many antibiotics and things in there. And the major one I'm going to talk today is about municipal wastewater treatment plant. That's my focus for today's talk. So what happened once CEC, why we are concerned about CECs? Once they produce, they go through various channels. They go in medicines, in veterinary, in manuring, and sewage, as I showed you various sources. Once they go all over the places in the soil, they all come together and leach out through the landfill or through various sources in the groundwater, as well as in the surface water through the flow. Once they end up in groundwater, they can be a source in drinking water. And there are actually papers published recently which are showing de facto water actually containing, and even drinking water plants actually containing CECs. That's why it's a concern and we care about it. There are a couple of papers which I just want to focus quickly through them. How many contaminants of emerging concern they found in the surface water through the wastewater treatment plant since 2005 to 2030. There are more recent papers nowadays, but I'm just covering the major one. They found, like, they did really large scale study about 90 wastewater treatment plant, and they found like around 80% of them. So it is a big issue. So what work has been done so far in my area and how I'm moving forward. So I will take you through my master's and then my dissertation and what I'm doing here in Brander Lab. So in first contaminant, which I picked was copper. It was in the two forms, copper oxide nanoparticle and copper chloride. And we tried to look what stress effects it's causing. I'm not showing stress data here. I'm just going to skim over their behavior data. And if you see, this is control group, and that's copper nanoparticle. If you see, pay attention to the ends, how they are curled up, and they are, so this one is brown, this is healthy, this is containing algae in it, that is in the symbiotic relationship. So it's providing food to the sea anemones. Here it is looking more transparent. So that means algae are leaving the sea anemone, and that's causing its color lighten, and they are ending up in the ends of the tentacles. In copper chloride solution, which was in its microform, that's the nanoparticulate form, its tentacles are all gone, and its color doesn't affect that much, but it can't function, it can't take food inside it. And that's within the 10 day of exposure. So you can see the stress actually from the images. Then we decided to see what is the climate change doing to these organisms. And for this, we use this PCO2 thing. As you all know, PCO2 is the partial pressure of carbon dioxide that's inversely proportional to pH. So for example, the carbon dioxide we add to the water actually lower down the pH, and that's what we use Henderson-Hasselbalch equation to create a ocean acidification effect into the lab. And that's what the system I used, where I created ocean acidification effect. And these are my replicas in the tanks. This is the system. This is the Loligo software system, which we were using to create the ocean acidification effect. In this top one, in the control, there is these two doesn't have copper in it, and all other had copper with different partial pressure of CO2 effect, showing different amount of CO2 we actually see in the system today, and what we are thinking of looking into in the next few years. So I'm going to go over behavioral observation. I'm not going through stress data in this one and results, or maybe later. So in this one, just within the 3 days at 400 ppm, that's our control group. It was not even, it can't even stand properly. It started falling off. When we added 10 microgram per liter copper, within day three, with 400 ppm of CO2, you see their tentacle started darkening. Their their base started lightening and their tentacle started shortening. So that's what it was causing within 3 days. At 1000 ppm CO2, it was studying properly, but it started losing its algae in there. And with 10 microgram per liter just of the copper with 1000 ppm, they just started losing tentacle and couldn't stand properly. So that was the effects we saw. Then we wanted to see what is happening in the nature within the macrophytes and algae in seaweed species. And what we found is we saw copper. It was absorbing copper so much into the seaweed species, and we just saw its accumulation effect, and we found a really good bioaccumulation effect in the seaweed. So it was causing effects from seaweed level to the invertebrate level. Then I moved through my dissertation work where my focus was, okay, then this is the contaminant of emerging concern in the surface water. How it is going to affect us as a human. For this, we took skip through aquaculture. And if you see, like through surface water, there are various studies which shows how much contaminants of emerging concern are actually in the fish within the aquaculture. And once they are taken by human, they can actually end up in our system. And that's what we try to show in this research here. Once wastewater with contaminants of emerging concern ends up into the surface water, it doesn't affect only water quality, it also affects the water quantity, because you are losing good amount of water and you are left with the bad quality water. That means you are leaving the quantity lower down. For this one, I prepared a model using R. The neural network I used to prepare this model, and I wanted to see how much wastewater treatment plant actually ending in the surface water. Here, this is for the continental US, and lower than 1 is actually shown with red. And the lighter the color, the better the water is coming. So gray water, that means it's really good. It is having less dilution or less concentration of wastewater treatment plant effluent. And as you can see within the US, this area actually has more concentration of wastewater treatment plant effluent, and it was partially because of the temperature, because of the population effect, as well as how much rainfall it receives. And these are the hydrological units we found actually affected by the wastewater treatment plant most. So what does it mean with the aquaculture? For that, I compared this data to aquaculture, and what we found is there was these polluted water actually ending up into the aquaculture system, which was used as a surface water. And just on a short note here, currently US is not using reusing water from wastewater in the aquaculture, but keep in mind that de facto reuse is actually happening, and this surface water is actually a part of the cycle within the system. So to identify, so far we know, like, okay, there are contaminants of emerging concern in the system. Now we wanted to find out if these contaminants of emerging concern are actually causing effect that can lead it to become a pollutant, or if so, how it is affecting the fish. For that, I picked up 2 compounds, diltiazem and PFAS compound. I'm going to discuss here PFAS only. PFAS is like a man-made chemical which was introduced in 1940s. And when we talk about PFAS, it can be found from non-stick cookwares, stain-resistant products. Firefighting foams are the bigger ones which are taking most attention nowadays because of the cancer issue in firefighters. It can be found in photography, pesticides, paints, and there are more than 6,000 chemical compounds actually belong to this family. PFAS came into existence from 1930s, where it was accidentally discovered by Teflon, and currently these are like actually facing so many charges and lawsuits, and there are constant pressure on the government to actually ban and limit these products because of their serious health impacts. So from the PFAS, I picked up Genex. Genex is a fluorinated carbon compound, which was introduced by DuPont in 2005 within the US. And it is to identify Genex, it is just it's a PTFE coated material on the pans, on the on the wire cables and the pans we use in the home. Genex took most of the public concern in 2017 and June when it was detected in Cape Fear River within the North Carolina city of Wilmington. And right now as well, it has most attention from that region because it was found so much over there. For this project, what I did is I took the fish fingerlings from tilapia. I exposed this to the Genex, 1 microgram per liter. Then I measured their bioconcentration and I see how much they are taking. I expose them with the chemical for 4 days and how much they are depurating because I want to see the aquaculture solution to this problem. For that, we kept them in the clean water and we analyzed data to get more analysis. So this is the semi-static flow-through system that was a randomly experimental system I designed for this project. And this is the base sump tank, which is taking 3 tanks that were randomly distributed with the tank system. So water is flowing into the system and then flowing out of the system to maintain the cycle. This is the real system looks like in the lab. For this project, we collected the liver, plasma, carcass, and fillet of the fish and analyzed it for bioconcentration factor, uptake and depuration constant, biological half-life, and tissue distribution. Bioconcentration factor actually tells how much a chemical actually going into the tissue of the fish. And biological half-life tells how long it is taking it to go away from the fish tissue. So these are some of the results here. The most bioaccumulation we found in the plasma, followed by liver, then followed by fillet, that's in the red, and then in orange in carcass. So fillet was the least one. And this is the uptake for 96 hours. It took like which one was the highest. And then they started following a lower trend when we kept them in the clean water. That was on the freshwater system. When we exposed them at 16 ppt saltwater system, a plasma actually uptake comparatively much larger compared to the liver, and then followed by carcass and then fillet. Fillet was the least one, and it also followed a pattern of depuration, but came to a steady state afterwards. And if we see them separately, carcass within the different salinities, we found like carcass doesn't have significant different other than few points at both the salinities. However, fillet was actually bioaccumulating more at the higher salinity compared to the freshwater system. Same in the plasma, we found higher concentration at the 16 ppt compared to the freshwater. With the liver, we found at some places, but not at all places. There were overlapping effects. So what we found is plasma has the most higher bioconcentration factor. That means it is bioaccumulating more after carcass and fillet, liver, carcass, and fillet. And comparatively, salt water has the more. However, fillet was most significant according to our results. In terms of biological half-life, we found fillet was actually bioconcentrating least amount of Genex in it. However, it was taking longest. So it took around 54 days. It will take around 54 days to actually removing out of the tissues compared to the liver. Liver was actually bioconcentrating more, but it was leaving the body really quickly at the same time. Carcass was the second one. It was taking 23 days. So what does it mean? Like even if we keep the fish in the fresh water just to depurate it, it is not economic to actually wait for 54 days in a fresh water. So all the tissue Gen-X going to remove out. In terms of mathematical calculation, what we found, so most of the time people, when they eat aquaculture food or fish, they focus mostly on fish fillet. Fish fillet at 96 hour in 16 ppt consists 0.312 microgram per liter of Gen-X. Once one fillet serving is actually around 3.5 ooze, that's equals to 100 grams. And when a person eats 100 gram of fillet, he or she is actually intaking 31.2 microgram of Gen-X. That means it's a single serving is enough to affect a person. So that was most concern for the consumer as well as the aquaculturist. We are working more to understand that situation and how to deal with it in our previous work. And now I moved to here at Oregon State University, and at present I'm working at Brander Lab, and we are trying to find out another contaminant of emerging concern, that's microplastics. We are looking at different microplastics. We are looking at thyroid particle. We are looking at polypropylene, polylactic acid, and microfibers. With all of these, right now I'm going to show you results from the thyroid particles because that's what I have right now. We are working more on on experiments and data analysis for all these things. But these are the contaminants of emerging concern right now, and we are looking more to understand what's going on. So for thyroid particle, we are working with myset shrimp and silverside larvae. So these are on a 30x scale, and they are 4 or 5 day post hatch larvae, and these are just 5 day post hatch larvae. We are working with 5 ppt salinity for silverside and 15 20 ppt for silverside and 15 to 15, 20 and 25 for myset shrimp, so different salinities. We are using 66,000 and 60,000 particles concentration for this work, and we are trying to find out how these particles actually affecting behavior, their cubic work, how much particles the fish or myset shrimp is actually intaking, how it is going to affect their growth. And later on, we are going to look into reactive oxygen species that will show how much stress it is causing in mycets. And in case of silverside, we are going to see how gene expression are changing through RNA analysis. For now, we are using NOLDUS as an instrument. This instrument helps us to identify how the fish is behaving in a system. We are using 3 dark light cycle. So for example, for some time it will keep dark and then a light turn on. It changes to this chamber changes to the light condition, and then we go back to dark and so on. And we are trying to see how a fish or a myset shrimp reacts when these things got changed. For so far, we found these results. These are behavioral results. I will take you a closer look how it looks like. So we are looking for the distance, how much distance a fish moved from the center, how many times it is freezing, what is their freezing frequency, how much it is freezing, and what is the movement, how they are moving, what is the in-zone duration, how much time they are spending in a zone, and what is their frequency, meander, and how much they are doing angle turn at various concentrations. So so far we found this is sorry. Okay, how to take it back? So yeah, sorry. So far we found in comparison to control, these they are actually showing significant results. So these 2 asterisk sign, and I'm going to take you through only 15 ppt concentration for microthyroid particle, and we found quite significant results from the control. Each asterisk is showing and P2 and P1 are the particle concentration, how much they are deviating from the control, how much they are actually affecting different from the control. And leachate is the solution where there is no thyroid particle after some time. So it has a leachate of thyroid particle, but not the actual particles in it. And you see fish is actually behaving differently from the control, and it is causing their health effects at both micro and nano level. For the mysid shrimp, before we have seen like similar effects where we saw a significant different behavior from the control at all micro, mostly at the micro level, fewer at the nano scale level. And that's where we are thinking maybe it's particle effect that's causing these issues, but we are still investigating what it looks like. Next thing was cubic work that was done by John Dickens in our lab. And what he did is he cleared the tissue from the post-hatch larvae of the mysid and silverside to see inside it how many particles it is intaking. And these results are from 60,000 particles per milliliter. And what we found is these thyroid particles were actually into the intestine of the mysid shrimp. It's here, the second one here, and this is much closer look how much this is their intestine and that's their stomach, how much they are actually they are taking into their body. So for the future direction, I'm looking into a combination of micro and heavy metals. Microplastics actually have a tendency to absorb heavy metals into their surrounding. I want to see with changing climate condition, especially ocean acidification, how these heavy metals are actually interacting with nanoplastics. With their interaction, how much are they going to bioaccumulate into the organism, and how they are going to transport to other parts of the ocean and actually shifting their nature from a micro heavy metal thing to higher levels, how they are going to transport to other parts. And using this result, I want to prepare a model that can help us to understand what the future looks like with climate change. And with that, I want to thank all my colleagues and my supervisors and my mentors who helped me throughout this research. And with that, I would be happy to take any questions. I think I went over time or over time.
All right. Actually, you have more time, but if there are any questions from the audience, please feel free to ask them. Let's see here. I'm not seeing any in the chat. Here we go. Okay. How did your Gen X concentration compare to environmental concentrations, wastewater effluent concentrations?
Thank you. My Gen X concentration was 1 microgram per liter, and that's environmentally relevant concentration. For so far, the limit imposed by North Carolina water treatment plant, they didn't impose it yet, but they are thinking is 17 nanogram per liter. That can be in the drinking water system, but they didn't impose it yet. So they were actually into the range, but it's still like we are trying to understand how much but it's like really environment friendly concentration.
Another question is what kind of aquaculture is is being conducted in deep South Texas? And they thought the shrimp farms in Harlan had closed.
So the aquaculture, I'm not sure much about aquaculture in deep South Texas because I was working more with the contaminants of emerging concern. But since it has a boundary with Gulf of Mexico, so I'm sure there is much more marine aquaculture promoted, and there are some freshwater aquaculture farms as well when I just travel through them. But I have no idea about shrimp farm in Harlan.
Another question we have in the chat is, can you summarize the biological implications of the behavioral changes in the tire particle experiment?
So, let's see what there. So, the behavioral, when we talk about, okay, sorry. Behavioral. So when we say behavioral in terms of biological, we connect it through neurological effects. So what an organism is thinking, how to react. When they react on something, they actually use their neurons to work on it. If it is not moving or it is showing freezing, that means it can't judge what's coming on to its prey, and they can actually die because of not reacting on the same time. So when we turn off and on the light, organisms didn't reacted the way they should be. That may have that have huge population level effect on them. So behavior actually have a biological implications. I'm sure I answered your question. If not, you can ask me like what specific you want me to ask.
I'll let Dave add any further questions to follow up. We have another question about TWPs. Are the TWPs TWP concentration used in your experiments based on real world concentrations?
Okay, thank you. Actually, 160 particle per milliliter is close to the real world, but 60,000 is not, because we want to see how it affects at higher concentrations. But if you follow, I know these lines are really like low color. I'm going to change them soon. But if you follow these P1 signs, actually lower concentrations also affect their behavior. For example, they freeze constantly. Their freezing frequency were really high at lower concentration. So yes, some of the concentration are environment friendly, but being a toxicologist point of view, we want to see what concentration actually affect them more, and we use 60,000 particles per milliliter.
So just to add on, are there any instances where the concentration would reach 6,000 or 60,000, like say industrial sites?
Look at those little bunnies.
Sorry, Jennifer, can you? Okay, sorry. Sorry, I missed your point. Can you please say again?
Well, so I was asking that are there any examples or possibilities of concentrations greater than 60 at 6,000 or 60,000, say at an industrial site?
Maybe, yeah, maybe. If they are industrial site, there may be, or thyroid particles, yes, there may be, but I didn't read so far. I read my lower concentration, what they found so far. It's a really new topic and new research area. Only few researches have been done so far on environment level. So we don't have that much data on it, but most of the concentration I found are close to the 60 particles per milliliter.
Okay. And those, so just those samples, do you have a sense of where those samples were taken?
Most of the research I saw is from the California, and it was a big topic of research, in the news recently. Most of the salmons they found died in the California region. When they investigated into the issue, they found that's the thyroid particle they were ingesting causing them to die. So that there were so many, that was the major one we found from the environment, but there are like papers from the other parts of the world as well, where they are seeing thyroid particle in the nature.
Okay. Thank you. We have another question online. Will future research investigate impacts of 6-PPD based on the results of work in Puget Sound that found this contaminant was causing high coho salmon mortality rates?
Yes, that's what we I'm planning to. Yeah, that's the one I was talking about, salmon mortality, which we recently found with thyroid particle. Yes, it's definitely, because thyroid particles are known to be correlated with zinc in them. So if they are there, that means zinc also there. It can help to understand like what is actually happening in nature. Are they dying because of thyroid particle or the metal into it? So that's the future research area.
Would you mind sharing, elaborating more on who you're collaborating with on that project?
Like in which one? In
this one, looking at coho mortalities and the effects of PPDs?
Oh, I'm not doing anything right now.
Oh, okay.
Yeah, so that's like that's another project that's happening in California. I'm saying I am planning to do something like that in my future,
Oh,
to correlate them to and see like what's going on, what is happening next.
Okay, great. Any other questions from the audience? Okay, well, it doesn't seem to be, we don't seem to have any more questions on the chat. Samreen, could I suggest that you post your email address in the chat so that folks will, and also you can see it right there on the screen, so that folks will have that if they do want to contact you. And Suzanne, do you want to add anything else since we're near the end of the seminar? Or Samreen, if you want to add any additional comments or thoughts?
Mm-hmm. I'll let Samreen go first if she has anything further.
I think I'm most of done.
Sounds good. Yeah, and that is a really good question about the 6-PPD. Those findings came out, I guess, in January, and that finding came out after we had already designed and run most of the thyroid particle experiments. And Samreen, correct me if I'm wrong, but we didn't see a lot of toxicity with the leachate. It was mostly the particles. Yeah. And I think I've been talking to others about this, and I think a lot of it has to do with how the extraction is done and the environmental conditions, might even have to do with salinity and species-specific sensitivity. So there's kind of a lot to unpack there, but it would be great to look into it in the future to see if we see a similar response with that compound with our model organisms as they did compared to what they did with coho. But yeah, it's a huge concern for sure.
Okay, well, if there's nothing, if there's no other questions, I think we will end our seminar early today.
Great. Thanks everyone for coming.
Yeah, thank you everyone for coming, and Samreen, thanks for giving a presentation today.
Thank you for inviting and giving me a chance to share. Thank you.
Bet. Welcome to Hatfield.
Thank you. Okay, bye.
Bye-bye.